Whenever a dancer leaps or a runner kicks off, their nervous system and body must work together to create a fluid movement. Every movement is supplemented with a variety of senses. For example, we can use visual aid, we can use proprioception to determine where our limbs are in space, and we can use cutaneous perception or tactile feedback to determine our environment. Just posterior to the central sulcus in the brain is the primary somatosensory cortex, or S1. S1 is responsible for a variety of those senses, including tactile sensation, proprioception, nociception, which is pain, and temperature perception. The motor cortex, M1, is just anterior to the central sulcus and controls voluntary movement. An effective neural prosthetic must be able to establish connections between M1, S1, and the prosthetic. However, before building connections between the artificial and the organic, we must understand how the brain communicates with itself. Neurotransmitters are passed between neurons to promote or suppress a reaction. These might open sodium channels along the membrane surface of a neuron. This causes the membrane voltage to spike in an action potential. After a certain point, sodium channels close, causing a repolarization. When the signal reaches the synapse, more neurotransmitters will be released. Neural prosthetics replicate action potentials with microelectrode arrays, MEAs, and intracortical microstimulation, or ICM. ICM are electrical pulses that trigger specific regions of the brain, mimicking the voltage spike of an action potential. MEAs are a series of electrodes implanted in the brain that monitor ICM. There are a few examples of some really interesting human trials. In one, a woman with quadriplegia was able to move a robotic limb with up to 7 types of movement, for example, rotating your wrist or extending your arm. She was able to feed herself a bar of chocolate by controlling her prosthetic with only brain activity. In another trial, someone was able to identify where pressure was being applied in a completely synthetic arm. In the latter trial, 2 pairs of MEAs were implanted, 2 in M1 and 2 in S1. These 4 working together enabled bidirectional control. Essentially, signals were sent from the brain to the prosthetic, as well as from the prosthetic to the brain. Normally, neurons tell a limb to go in certain differing directions. When these overlapping signals are pooled together, one signal is sent to the arm to tell it where to move. In a brain-computer interface such as a neural prosthetic, equations are used to describe the relationship between neural activity and movement. Using feedback from MEAs recording signals in M1, movements can be artificially executed. Evoking sensation is a bit different from creating movement. Parts of S1 correspond to different parts of your body. For example, one region might be responsible for sensation in your hand or your forearm. Specialized sensors on the prosthetic act as nerve endings and send signals to the brain, or in this case, the MEAs. When the signal is received in the MEA, the electrodes stimulate the appropriate part of S1 that corresponds with the part of the limb that was originally stimulated, evoking a touch sensation. Who knows where this technology will go in the future? Ultimately, it has a lot of room to grow. It's quite expensive, movement isn't very naturalistic, and tactile sensation is limited. However, it is growing. Every year brings new advancements, and every year we get smarter and better at using brain-computer interfaces. Maybe someday we'll see a future that successfully integrates man and machine at a commercial level. Thank you.